Hold on. Series. My great pleasure to introduce Stefano Fusi today. Stefano is a professor in neuroscience. I'm not going to go through all the titles, so many of them don't forget. He's an investigator at the Columbia Zuckerman Institute. I'm also a member of the Center for Theoretical Science. Now, I looked at Stefano's career and training today spans multiple countries: Italy, Israel, Switzerland, and the US. I found one interesting fact that his interest in theoretical neuroscience started while he was a researcher at the Italian Institute for Neuroscience in Rome. I'm guessing that's a very interesting story, like how this judgment in theoretical neuroscience was. But today, Stefano is a leading computational and theoretical neuroscientist, and he's working with many corners of our field, from all the way from single neuron and network dynamics, to memory and learning and data representations. He's really exploring fundamental trade-offs between abstractness, expressivity, and robustness in neural representations, basically also being very close in time to neural data, which is impressive, and that's what we're going to hear today. But it's a great
Thanks a lot, Janice, for the introduction and for the invitation. I'm really having a great day. There are so many fantastic people working on wonderful projects. And so what I'm going to talk about today is the geometry of abstraction. I'm going to define everything.
You know, the idea is that basically when I started to
switch from physics to neuroscience, I realized that the brain is really a mess. You know, if you look inside and you start analyzing the data, it's so difficult to find interesting patterns, something that is highly reproducible. And I thought at the beginning it was really hopeless, but then I started to realize that there are actually some structures that are interesting for several reasons. And in particular, there are some that we observe in a lot of different brain areas across different species.
So in particular, the type of structure we were looking.
Well, some kind of implausible structure, to use the word of Ben Harris. It was at a conference recently. We were discussing about this. So, you know, it's something that you would not see by chance very easily. So something that is unlikely to observe by chance. Moreover, this kind of structure that is summarized by this geometry of abstraction is interpretable and it has a computational. And finally, it seems to be correlated to the behavior of the animal. So, you know, that's the first indication that it might be something.
Okay, so today I'll start from the first studies that we published 5 years ago. And then I will go over the more recent
discoveries across multiple different species. So I'm going to consider rodents, monkeys, and humans, and many different brain areas. Now, try to identify some of the patterns that are small preserved, are more meaningful across all these different species.
Okay, so you know this project.
The emergence of people. Here I have a partial list of all my collaborators. I'm not going to go over it now, but later, you know, what's the exact contribution of each of these persons.
Okay, so since we are looking for structure in neural, first thing that I would like to do to talk about the structure. What do we want to structure?
What I'm looking for in the neural activity, it's certainly something that might reflect the structure of the brain.
And I'll try to tell you in a few words what we are doing, simply by looking at this very simple example.
So here you see a scene that you can immediately describe using very few words. I don't know if you look at this, you immediately realize that there are only two shapes. Now there are cylinders and spheres, and only two colors. You know, they are placed in different locations, They have different sizes. Some of them are stackable, like cylinders, some others are not. But when you look at these representations of the pixel level, these are really complicated. So, this is millions of pixels in a video, and it's extremely difficult to say that there is something interesting.
But as soon as you look at the whole picture, then you can use language to
describe this in a very few words.
So what kind of structure do we have here? And how do we expect to observe this kind of structure in the neural activity? Okay, so let me simplify even further the scene. So now I'm focusing on 4 objects. So one thing that you can start doing when you look at this kind of words is to identify the regularities. And in particular here, you might realize that these 2 shapes are similar. Okay, so these are 2 cylinders. And one thing you can do is to put these 2 objects into the same category. So the category is just a collection of items that are grouped together as equivalent for some parts. Now, the fact that you're putting them together might be dictated by their aspect, like in this case, or it might be dictated by their semantics. For instance, the cylinders are stackable and the cylinders are not. So that's something that is not directly related to the visual aspect, the sense, but to what you call. So very often in cognitive things, that's what we do. We group together things that look very dissimilar, but they have a similar meaning or they're used in a similar way. Okay, so now the concept is just a mental representation. And that's what I want. Here, for instance, we can introduce the concept. But we can also group the other 2 objects together into another concept, which is sphere. Now, this is an interesting example because it shows one of the important properties of the structure in the real world. And that is that each object can be associated to multiple concepts. It can belong to multiple groups. So for instance, here I can group together these 2 objects because they're blue, or these other 2 objects because they're yellow. And so I have other 2 groups. And then when I look at one particular object, that will be associated to what is called the concept vector. That is telling you whether this object belongs to a group or it doesn't belong to a group. This one belongs to the cylinders group, doesn't belong to the sphere group. Okay, so how do I represent kind of structure in the brain and in the neural activity? So here, let me just tell you one more thing, which is that in this particular case, the group of cylinders is mutually exclusive with the group of sphere. And essentially what I'm doing when I go from one group to another group, I'm changing something, which is what I'm going to call shape. Okay, so this is a variable. And in the other case, when I go from yellow to blue, I'm changing. Okay, so now let's look at the brain. So please interrupt me anytime if there's anything unclear. I really want this to be as interactive as possible. Okay, so now I'm looking at this space. So, I'm going to spend a few minutes about this because this is the space that I'm going to show you a lot during my talk. So this is the activity space in the brain or in an artificial neuron. Along each coordinate axis, I have the activity of a different neuron. Okay, so this would be neuron 1, neuron 2, neuron. I'm showing you only 3 axes because that's all I can do here on the slides. But you have to imagine that this is embedded in a very high dimensional space where you have thousands of axes that correspond to thousands of. So one of the hardest things is to try to imagine this in a high dimensional space. Okay, so now imagine that you are in this virtual world that I showed you previously, and that one stimulus is shown on a screen. And then you record the activity in response to this particular visual stimulus. So that is going to be a vector with many components. Each component is the firing rate of a different neuron. And that will be a point in this. Okay? So this is the population response to a particular space. Now, every point that I'm going to have in this space is going to be the response to a different stimulus. Of course, that will be presented at a different. Okay, now the first thing I want to do is to construct a representation that clearly groups together all the same shapes. So I want to have all the spheres that are grouped together and all the cylinders that are grouped together. And I'm going to literally do that. Okay, so this is the simplest possible representation in which I represent this concept. Okay, so all the cylinders are on top of each other, all the spheres are. Now, this is what I would legitimately call an abstract representation of shape, because it's entirely disassociated from the specific. So here, whether it's yellow or it's blue, it doesn't matter. I have exactly the same. Okay, so it's invariant with respect to this other feature, which is the same for this. Okay, so this would be compatible with the notion of abstractness that many of us have. Now, this is a bit of a waste of resources. Here I have thousands or hundreds of thousands of dimensions, and I'm basically constructing a representation that is 1D. And the only information I can represent here, if I take this literally, is only the information about shape. Okay, so I can do better than that. So for instance, let's say I consider now this representation. I have that the 4 different objects are now represented in a way that the population responses are arranged on the vertices of a space. Yeah, have this low dimension, relatively low dimensional structure. Okay, now this representation has also the interesting property of reflecting some process of abstraction, because now I have that shape is represented along this direction and color is represented along this other. If I project these 4 points along this direction, I get, again, a representation that is invariant with respect to color. Okay? So this is an abstract representation that is totally equivalent to the one that I constructed in the first example. But at the same time, here, if I project along a different axis, which is orthogonal to the first one, I get an abstract representation of color. Now, this is invariant with respect to shape. So I have abstract representation in subspace. If I project, then I get this invariant. Okay, this is a very interesting type of representation, and it's called with many different names depending on the community. So, machine learning community, many people would call this disentangled representations, factorized under some at the level of responses of individual neurons. It's what Doris Sau would call the axis code. I would call it linear mixed selectivity, because here essentially each neuron responds to a linear combination of shape and color. But it has a lot of names for a good reason, because it's an interesting structure. It's also not obvious. Okay, so now this is an interesting structure. I constructed it by hand, and now let's say I'm given some real data. The real data never looked that clean, and what I could get is an arrangement like this. So now, essentially, the set of all the distances is what defines the geometry of the representation. Now, this arrangement of these points, it's not clear whether I could call it abstract or not. So now, more precise definition and a more quantitative definition. So let's go back to the initial example. Now, why do I care about whether a representation is abstract? The real reason why I care about it is for its computational impact. It's for the fact that actually this kind of representation allow you to generalize in a non-trivial way. Okay, so this is actually a really simple example. Let's say I'm going to train a very simple linear decoder only on the blue shape, okay? Only on the blue cylinder and the blues. Okay, so only these two. So now a linear decoder would be an hyperplane in general. In this simple case, it would be a plane separating the cylinder from the sphere. Now, because of this arrangement of points, this would work right away also for the yellow. Okay, so if I'm going to test on these other two, of course it works. Okay, this is really a trivial case, but this is what we call cross-condition generalization. And if you think about it, it's already an interesting form of auto-distribution generalization. Okay? Because this decoder here has never seen any yellow object, but it works right away thanks to this. Okay, so now let's say this is really what I care about, is the ability to cross-generalize, which is, again, another non-trivial problem. So now I go back to this. And it's interesting because also in this case, I can play the same game. So I can train a decoder on these two objects, and this would actually work right away also on these other two. Okay, so this cross-condition generalization. So this is what I'm going to use as my defining characteristic of an abstract representation. So essentially, every time I compute, it has this strange acronym that Marcus Brenner came up with, now we're stuck with it, the cross-condition generalization performance. I will measure it for my geometry. Okay, so this is the nice and clean and very simple geometry, but it could be any geometry. I'm going to measure it. And I want to know whether this cross-condition generalization performance is significantly larger than the one in the case in which I don't have any structure. So what I mean by that? What I mean is that I take these centroids of, typically you have a cloud of points around each case, and I place them at random locations in my activity. So if I do that, I get an object that has the maximal dimensionality. And in this particular case, with these 4 different points, it defines a t-trait. So now this is a 3D object that basically has no structure because the positions are at random locations. And I can measure the cross-condition generalization performance for this one, and it's very close to it. If this cross-condition generalization performance for your data is significantly larger than for this null model, then I would say that my representation is abstract for that particular variable I'm testing. Okay, now, why is this implausible? That it's unlikely that this happens. The reason is the following, that here I have one very specific property of this geometry that is not true, which is the parallelism of this coding direction. So this is the coding direction of shape only for the blue shape, and this is the coding direction for shape only for the yellow shape. Okay? And they happen to be parallel. Now, this is not trivial, because when you consider high dimensional space that I cannot show you, then you take 2 random vectors, like in this case here. These 2 directions will be typically orthogonal. So the 2 vectors will be orthogonal. Okay, so if I take a very high dimensional space, I have thousands of neurons. This will typically be always orthogonal. And if I see parallelism, something interesting. It's something that's worth investigating because it's probably denoting, first of all, some non-trivial learning process, and or it indicates that there is already some interest indeed. Okay, so my advice is always look for parallelism. Here, it's also interesting that this direction is orthogonal to this, but this is not that diagnostic when you're analyzing the data, because again, orthogonality is something you get for free in a high dimension. Focus on the parallel. Okay. Now, so the first thing I need to show you is that we see these implausible representations. If we don't see them anywhere, then it's not an. So it all started with this paper that was published now 5 years ago in 2020. So this is a collaboration with a group of Daniel Salzman at Columbia and Silvia Bernardi collected the data. Marcus Ben and Mattia Rigotti, they developed this conceptual and theoretical framework, and they did the analysis. Okay, so let me tell you briefly about the task. There are several reasons. The first one is that it's an interesting story, but it's also because the same experiment has been replicated in humans, and I'll tell you what happens in humans in the following slide. Okay, so the trial starts with a monkey that has to hold the bar, and then a stimulus appears on the screen. It's a completely meaningless stimulus. It's a fractal. And the monkey has to learn that in response to this particular stimulus, it has to release the bar. And if it does so, it receives a reward. Now, there are 4 different stimuli that I'm going to call A, B, C, and D. And for some of the stimuli, the monkey has to release the bar, A and C. And for the other 2, the monkey has to keep holding. In these 2 cases, the monkey is going to receive a reward. In these other 2 cases, it just avoids punishment, which is a type. Okay, so there are 2 different required motor responses and 2 different possible outcomes that are orthogonal to the motor response. Okay, now the interesting cognitive component of this task is that at some point, without any warning, so this is important, there is no cue, they change the rule. Okay, so the rule now is with the same stimuli, exactly the same stimuli, a different set of visual motor associations and a different set of outcomes. And then these 2 task sets, they essentially define 2 different context 1 and context 2. And then they would go back and forth between these 2 contexts multiple. Okay, so these monkeys are really overtrained on. So they know that in this world, they can only be in either context 1 or in context. Keep going back and forth, multiple sessions for thousands. Okay, so now if you understand this rule, then you can do some interesting form of inference. So when you switch from context 2 to context 1, as in this example, so this is a sample sequence, the very first time you are presented with a stimulus in the new context, you cannot know that we changed. Okay, so there's no cue. So the monkey is going to respond according to the previous context. But now that the monkey knows that this rule is not valid any longer, it can switch right away to the alternative rule. And actually does it. So, you know, this is the very first time now A is presented in the new context after this switch, and it can already respond. And actually does it. So this is the data. So this is the performance right before the switch. I have a sample sequence down here. So this is the very first stimulus. The monkey cannot know that we changed the rule. So it's more or less 1 minus the performance for the context change. And then, you know, this might be repeated twice or once, doesn't matter, but this is the very first time you have the occurrence of stimulus A after the switch, after this particular switch, and you see that the activity is already above chance. Now, this is the very first time you present the 3rd stimulus after the switch, and it's even higher, and for D is basically the asymptotic level. Okay? But still, remember that this is the first time that you see this stimulus after the context switch, this particular switch. Okay, so this is something you cannot do if you just relearn the visual motor association. You really must have an understanding of the fact that there are only 2 contexts in the world. And then you just need to switch from one. So it doesn't go to 100% right away because monkeys make mistakes from time to time that are not due to the fact that we changed the rule, just the fact that they get distracted. And so, you know, they don't switch immediately, but we have evidence that they really switch at some point. This just reflects the average of the switching. Okay, so now let's look at the geometry. So I'm going to go very quickly about this because this is a relatively old paper and I want to focus on the other job that we found more. Okay, so this is one of the very few cases in which you take the activity from thousands of neurons you project it down to reduced dimensionality space, and you already see some interesting. Now, we did all the analysis in the original space using cross-validated quantities, but again, this is really one of the very few cases where you already see the structure, and that interesting structure is basically a cube. Okay, so let me show you here. I have the 8 different conditions. So here, every point corresponds to one of these 8 different conditions. Okay, so the first label here is context. Then I have the letter that indicates what stimulus is presented. Minus means that then there is no reward. Plus means that there is a reward. And then this is the result. Hold or release. Okay, so now this is very nicely organized. Not only it's a cube, but you see that context, all the 4 points that correspond to context 1 are here on the upper floor of the and all context 2 are here. Okay? So context is represented along these vertical directions, and these coding directions are approximately parallel to each. Value is represented along this direction, is approximately orthogonal direction. You see the front face has only pluses and the back face has only minuses. And finally, action is represented along this. Okay, so it's a beautiful disentangled abstract representation. And it's also abstract in a quantitative sense, because if we measure the cross-condition generalization performance, and we do it for all possible dichotomies that correspond to different variables. So, if you divide, let's say, the 8 conditions into 2 groups, context 1 conditions, context 2 conditions. This is one particular dichotomy that corresponds to context. I can also divide the points into this group here and this other group. So that will correspond to value. So these are all interpretable variables. But I could also group together, let's say, this point, this point, this point, and this point. That probably doesn't have any name, but who knows what this monkey is representing. So we wanted to do the analysis in a completely unbiased way. Given that we have only binary variables, we can do all possible combinations. And every dot here corresponds to a different dichotomy or a different possible value. Okay, so this is the analysis. This is what we get. And you see that basically 3 that are really high, and they correspond to the dichotomies that you would expect by observing this geometry. So the top one is value, second one is action, and the third one is. And these values are basically different from what you would get in an unstructured geometry. Okay, so now this is the representation that we got. In the anterior cingulate cortex, we see something similar in the hippocampus and prefrontal. Looks like.
What percentage of the variance does this geometry capture?
So the 3 dimensions?
Yeah.
Yeah, so again, as I said, all the analysis is done in the original space. So the CCGP is computed in the original space, and it takes into account also the noise. But if you want to know how much of the variance this captures, I think it's about 80%.
It's not bad, but we know situations in which even if you capture 80% variance, the remaining 20% is really important because it's what makes the difference between correct and incorrect values. So never rely on these plots, but I'm doing it because it's very instructive to look at them, but the analysis was all done in the original space. Okay, now, one of the things that we didn't know at that time is how this representation forms, because it takes months to train a monkey to do this.
There's no report. The second thing is,
how do we know that this particular aspect of the representational geometry is actually relevant to perform the task, to do inference? Okay, so I started to work together with
the team of Ueli, Ruthshauser, in particular Yuri Minja, who's here, is the one who started this project, and then Christos completed it. And this is an experiment we did on epileptic patients who volunteer to do this kind.
Okay, so they have some severe form of epilepsy, so they implant these electrodes for a relatively long time, and then do this study. Okay, so this is a pretty amazing data set. I don't know how much you're familiar with this kind of studies, but they have something like 3,200 well-isolated neurons. And I remember in 2014, you could see articles in Nature Neuroscience with 4 or 5 neurons recorded from this. So this is 3,200 neurons across many different brain areas. Okay? Campus, the amygdala, PSMA, ACC, and ventromedial prefrontal cortex, and basically. Okay, now the experiment, the protocol is exactly the same. So there are 4 different stimuli, 2 possible responses. Now, they have a keyboard, they can press left, right, and the outcome is a monetary reward, 25 cents or 5. And again, without any warning, at some point, switch from one response to another, the one task set to another. So humans can also do inference. Okay? So this would be essentially what we define as an inference trial. So it's the very first trial after a switch and a mistake. Okay, so now subject knows that it's time to change rule. And so, humans are really good at doing this, and they have almost 100% performance immediately after. Now, what do we see in a session in which they can actually do it? So this is the geometry, and the geometry is very different from the one that we see in monkeys for many reasons. So one of the reasons is that humans learn this task in one session. And also the humans, you cannot choose where you record from, so there is only some partial overlap between the brain areas that are targeted in the monkey and those that are targeted. But anyway, the kind of geometry you see, so when you consider one concept,
the stimuli don't really have any structure.
so you expect them to be at the vertices of a tetrahedron. And that's basically what we see. Now, I'll show you a cartoon, and then I'll show you the real representation. Okay, now, when they can do inference, you have to imagine this in 4 dimensions. That's the minimal number of dimensions you can describe this geometry. So now, with the blue line, you move along the 4th dimension, and you go from one context to another context. And then you have another tetrahedron that is very similar to the first. It's just shifted, where you have the second. Okay, so this is great because all these coding directions for context are parallel to each other, and also for pairs of stimuli. So if I go from A to D in context 1, this is parallel to A to D in context 2, and analogously for all the other pairs of stimuli. Okay, this is a highly non-trivial geometry that we observe. And now I'm going to show you what happens at the beginning when humans still don't know that. So in order to tell you what happens during learning, I would have to show you this in 4 dimensions, and it's not that clear. So what I'm going to do is to consider only a subset of the stimuli. I am going to exclude one stimulus, so now I can more easily visualize it in 3 dimensions. So I would expect one triangle here, another triangle that is aligned. Okay, so this is real data now, again, projected into 3 dimensions. All the analysis is done with cross-condition generalization performance, and another quantity called parallelism score that I'm going to mention also later. But this is what you have at the beginning, or essentially in the non-inference. So these are sessions in which the subject still cannot do inference. They can perform the task, but they cannot do inference. So basically, they're really good at the end of a block of trials in one context, but they cannot switch right away. Okay, so many of the quantities that are relevant are encoded. You can decode them using a linear decoder, but you have this messy geometry. Now, let's see what happens when you're learning. Okay, so this is the transformation when you go from a non-inference to an inference. Okay, this is combining together multiple subjects. I should have said that, but otherwise it wouldn't be so. So you go from something that is highly disordered, okay, this one here, to this beautiful structure that I'm going to show you again, in which now you see that the coding direction for context, so this one here, and this one here become parallel to each other. And also this direction for pairs of stimuli are parallel. Okay, so now let me summarize all the changes. I mean, this is all published, so you can read all the details of the analysis. I'm not going to provide you any. But again, all the analysis is based on cross condition. Okay, now there are a few things that change during learning, and it's interesting because this is really one of the very few cases where we can monitor the formation. Okay, so first of all this happens only in the hippocampus. No other brain area that they record. The second thing that they observe is that the average activity significantly decreases during learning. Now, this has some implications for the geometry. In particular, that all the distances are going to shrink because the firing rate will go down. So it is true, actually, all distances shrink except for the distances between two constants. So this distance actually increases slightly. No. Whereas all the other distances decrease. Okay, we analyze also the noise in this case, and we see that the noise decreases, which is another possible explanation for this better ability to generalize across concepts. But the noise decrease is entirely explained by the firing rate reduction. You reduce the firing rate, you assume a very simple Poisson model, you're going to get a decrease. And this, yeah, the noise decreases entirely. Now, the most interesting transformation is really a geometrical. And it's the fact that the coding directions for context, so this one here, and pairs of stimuli, so for instance, this one here and here, they become more tight. So initially, they are basically orthogonal, and when the subjects can do inference, they become parallel. Okay, now, they recorded from 19 different. Some of these subjects are relatively old, they might have dementia, and they just don't learn in one session. So what they do after one session is that they give them verbal instructions. They tell them, these are the rules of the game. Now they can do that. But many of them can do the task and they can do inference. So, this is the case in which they start without having the ability of doing inference. So again, the performance is pretty high, as you can see here, just before the end of a block of trials in which you are in one particular context. And then when you switch to the other context, you see before these verbal instructions, they're not as good as after. So after the verbal instructions, they make one mistake, but then the very first time they're presented with the second stimulus, they're basically 100. Okay, so some other subjects, they do this already in the first session. And finally, there are subjects that never. Okay, so what happens now to the geometry of the rock? So when I look at this, what is relevant for me is the parallelism between these coding. So we define what is called the parallelism score. So it's basically cosine similarity of these vectors, of the coding directions. And it's compared to the proper null model, but essentially what we get is parallel. And this is before giving the verbal instructions. This is after. Okay, so if they can do inference already in the first session, where they can only learn by trial and error, like the monkey, the parallelism score is already very high. They receive verbal instructions, it remains the same. Okay, if they cannot do it before or after, then the parallelism score remains very low. But if they cannot do it in the first session here, but they can do it in the second session, you see that the parallelism score goes up and actually becomes very close to the parallelism score of the subjects that learn by trial. Now you can look more carefully at many different aspects of the geometry, and you see that the geometry that you observe after the verbal instruction is incredibly similar to the one that the subjects learn by trial and error, in the same way. We don't know why, and if you guys have any ideas, that would be really something interesting. But it was an interesting observation. And there are so many people among you working on large language models. Okay, so now I'll go over many other geometries to give you many different examples, and I'll try to make an effort to give you what is my impression about what we learn from all these different studies. Can we really have a few take-home messages that now look stable enough that I feel comfortable to talk? Okay, so another interesting abstract concept is familiarity, and it's a beautiful example of an abstract concept because it can be applied to so many different domains. Okay, I can ask you, whether these faces are familiar or not. I can also ask you whether this place is familiar or not. And it can be applied to objects and to many other different categories. And it's something that we can do right away. Even if I introduce completely different type of objects, you can tell, yeah, this is a novel object. Okay, so now how is familiarity represented at the neural level? Okay, so the first study I want to discuss is something that has been performed in the laboratory of Sigelbaum, Colombia, and Lara Boyle did all the experiments, and Lorenzo Posani did the analysis, the simulations, and he also developed this theoretical framework. And by the way, Lorenzo is a fantastic postdoc who's on the job market. So if you need to hire a theorist, is really probably one of the strongest candidates. Okay, so now this is an interesting task because there is a social component, and you see this mouse. This is the subject mouse. It goes around in this arena. Now, I don't know if you can see them, but there are other two mice under this pen cap. And this mouse can go around, interact with them, and explore the environment, spend some time with these mates. And they did the experiment changing a lot of these variables. So let me first list all the variables that are relevant. Okay, so there is the position of the subject mouse. And let's binarize it. It can be close to this animal or close to this other animal, so left or right. There is another variable, which is the mouse identity. Okay, so here I have one mouse, here I have a different mouse. Now, you already have a confound here. Okay, if I find selectivity to this location, is it selectivity to the position of the animal, or is it selectivity to the mouse identity? So what they did was to do all possible combinations. So they did the experiment this way, then they swapped the position of the two animals and repeated this multiple times. And there is also a third variable, which is the degree of familiarity of the mice that you put under this. So again, it's another binary variable. These animals can be highly familiar, they are litter mates, or they can be completely not. And again, they do all possible combinations. So here there are really a lot of experiments that have been performed. Okay, so let me summarize the geometry. Again, you're going to find a lot of plots with CCGP, and so on in the paper. but this is what you get. Okay, we were very surprised.
Now we're in the hippocampus with differentiate2, which is known to be important.
Social memory. When you look at the representations in response to the interactions with the novel animals, you see this beautiful disentangled representation. This is really flat. It's the flattest representation that we observe. Identity is represented along one direction and position is represented along the other. Position is more strongly encoded, so you see this distance is larger. Okay, now let's see what happens when you switch from novel animals to littermates, so that they are very familiar. Okay, so there is this transformation. Everything is shifted up, and it's also distorted. Now, there are a few things here that we should. So first of all, these two variables here are disentangled, only for the novel animals. They are not for these familiar animals. So this nonlinear. However, all these coding directions for familiarity, they are parallel to it. This is a very strong signal. We see it everywhere in the brain, this familiarity. And in this particular case, all these coding directions are parallel to it. So you train a decoder to report familiarity for, let's say, this particular stimulus, this particular mouse at this location. That would work right away for many other mice and many different locations. We did that for 4 different mice. So it can generalize very nice. Now, apart from this shift upward, there is also a slight compression along the identity axis. This is a bit surprising. Now, these are familiar mice, but they're less decodable than the novel mice. And you definitely have nonlinear distortion that make this representation high dimensional. You start from a low dimensional representation here, and then with learning in the hippocampus, you get this higher dimensional representation. So this is nicely compatible with many studies that show that you decorrelate the stimuli in the hippocampus and you make the representations right. But why do we want this nonlinear distortion? Okay, so if you simulate a very simple network, you start from a latent space, you have a certain number of small number. Then you generate a high dimensional representation, to be the word representation. And you pass it to this very simple recurrent network. Let's say something like an option. And then you want what is the storage capacity for this network. You can do this actually analytically. And then what you see here for this representation is that as you increase the distortion and you go from this very low dimensional representation to a high dimensional representation, cross-condition generalization performance, of course, goes down, but the storage capacity goes. It's not too surprising. The storage capacity essentially scales with the dimensionality of the representation. But it's nice that we can observe this in the hippocampus. And it's also interesting that the representations initially are so low dimension, which is definitely something that you must have learned, but you probably have learned that over many more experiences. And then, it's your initial condition only in this particular experience. Okay, so now, next, we were wondering whether this geometry is affecting in any way the behavior. And we want to find this relation. Again, it's just a correlation, but it's an indication that it could be important. Okay, so if you take a mouse and you put it in this arena, and you have a familiar animal and a novel animal, the mouse, unlike humans, is going to spend more time with the novel animal than with the familiar. So this is something well known. Now, let's look at the geometry. I want to characterize the difference in the geometry between this representation and this representation. I could measure the dimensionality, but it's actually, I can see a stronger change if I focus on the decodability of one particular dichotomy, which is essentially the exclusive. So if this is completely flat, then you cannot separate these 2 points from these other 2. So here, I cannot find an hyperplane separating these 2. But in this other case, I can. So here I'm showing the social preference score on the y-axis and the difference in the decoding ability of the exclusive OR between litter names and novel names. So these are different experiments, and you see that there is a very nice correlation. So basically, every time you see a higher dimensional representation, which means that the decodability of the exclusive OR is much larger, then you see a higher preference score. So the animals spend more time with the novel animal than with the familiar. Everything is balanced, of course, we balanced all the time intervals, number of neurons, and everything. Okay, so this is another example in which we can show a nice correlation between one particular aspect of the geometry and the behavior. Okay, now let's go to humans. So this is a different study. It's again from Ueli Rutzhauser lab, and Yuri Mincha was. And in this case, they were recording from multiple brain areas. I'll show you in a second a couple of examples. And this is the task. So there are actually 2 different tasks. So there is a block of trials in which they have to perform a memory task. Have you seen this image before or not? And a categorization where they have to say whether the image belongs to a particular category. Also, the response can change, but this is not going to play. This is presented, and subject has to respond. Okay, so now this is the kind of geometry that we get, and I'm highlighting different variables. But now focus on the red line. The red lines are the coding directions for familiarity. And again, you can see that they are parallel to each other. This is a very nice geometry. It's also so nicely organized because you see that along this other direction, the blue direction here, you go from one task to another task, and then task type and category is along this other. Okay, so you might say at this point, well, I mean, you see it everywhere. So is there a place where you don't see it? And actually, yes, there are a lot of places where you don't see this kind of representation. So for instance, this is in ACC. And you see these kind of representations where familiarity is definitely not up. And you can do it more quantitatively, but this is just to show you what
Familiarity here is only in not.
That doesn't mean that you cannot decode it. You can decode familiarity, but it's not in this particular case.
Does this analysis require binary categorization, or have you tried it on non-binary things?
Does it require what?
Binary categorization.
Yes. So in our case, to have
A very clear null model, we binarize all the variables. And whenever we have multiple values or continuous value variables, we binarize them, or we consider one pair of values at a time, and then we do the other. But we are developing tools to do the analysis in all these other cases direct without binaries. Yeah, but binary is really robust. It's very easy to do there. Okay, one more example about familiarity. So this is something, a collaboration with Doris Sau, and it's a paper that came out recently. And Liang She did all the experiments, and Marcus Bedna now running his own lab at UCSD with the analysis that I'm going to refer to. Liang She also did a lot of. Okay, so they have recordings from these face patches, and they are interested in face familiarity. So again, there is a social component. So these are the responses of neurons to many different categories of visual stimuli. And this is just actually to tell you where they recorded from. So they recorded from AM, which is the anterior face patch, from perirhinal cortex, again from the face patch, and from the temporal pole face patch. Okay, so now here we see something again that is very interesting because the geometry is dynamic. It changes during the trial. So at the beginning, the initial response, we see that familiarity is already in an abstract format, weakly encoded, but it's there, certainly decodable. And here you can see these beautiful disentangled representations that Doris has observed already a long time ago using this axis code, in which you have some linear combination of face features that are represented along orthogonal lines. Okay, so now this is the initial response, and familiarity becomes decodable different times in different brain areas. It's reported 95 milliseconds in AM, 105 in perirenal, and 135 in. Okay, the late response is very different because in this case, this coding direction is parallel to this. Here, the code changes. And for familiar faces, it's this form. Now, we actually don't have enough data to tell whether it's non-linearly distorted or linearly distorted, but it's certainly distorted in the sense that this coding direction is not parallel to this. However, these coding directions for familiarity are still approximately parallel to the point that the cross-condition generalization for familiarity is pretty high. So again, familiarity, this is another example in which it's encoded in this abstract. Okay, now let me summarize what I showed you about familiarity. So familiarity is represented in an abstract format in the hippocampus, in rodents and in humans, and in IT perirenal, not in AC. So the identity of the individuals is more decodable for familiar individuals in the cortex, but not in the hippocampus. You also have much more detailed representation than in the. We definitely see nonlinear distortions in the hippocampus of rodents, not in the medial temporal lobe of humans, but they're performing two tasks, so we don't know exactly whether they're comparable or not. And it's not clear in the data from Doris South lab. So you see that there is clearly one big issue. And the main problem is that we are learning a lot from this analysis, but it's very difficult to draw conclusions that concern the organization of the entire brain. And the problem is that all these experiments have been done in different species, in different animals, and in different experiments. The experimental protocol is different. So, that's definitely a fundamental problem. The only thing I would be confident to conclude here is that these abstract, disentangled representations are certainly observed in multiple species and in multiple brain areas. So it's an interesting structure. Look for it, because you will probably see it, and that can teach you something, like what we learned about the hippocampus and the fact that there is this transformation in terms of the geometry. Okay, the other thing is that the geometry correlates with behavior. And so maybe, this indicates that it could be important. And our dream is to manipulate the geometry and see the effects on the behavior. But, we are probably 10 years away from being able to do something. So we have some ideas about how to do. Okay, now there is another thing that we could start doing. So there are data in which people recorded from many different brain areas, and the animal always does the same. Now, if you can do that, study this kind of systems, maybe you will realize that there is another property that we observe a lot in all these data sets. I'm going to summarize here. It is the following. So, this kind of representation has this high cross-condition generalization. It's disentangled, it's beautiful, low dimension. But there is one thing it cannot do. So if you want a linear decoder to separate these 2 points from these other two, you cannot do it because this is a typical exclusive OR situation and it's non-linearly separate, as I showed you. Okay, now you might say it's not a big deal. This is just one dichotomy, and who cares if you cannot do it? But if you increase the number of points, the vast majority of dichotomies will have this property of nonlinear separability. So you have to deal with it. Okay, now the interesting thing is that if you start from this representation, so now you start from a sort of a scaffold that is low dimensional, and you introduce some distortion, okay? Even a relatively small distortion. I mean, it should be larger than the noise, but you have a distortion like this, then you can separate these 2 points from these other 2. Okay, now for this kind of distortions, you can still have a relatively high cross-condition generalization. Okay? It's not perfect, but it's not a chunk. So it's still an abstract representation. Okay, now this is a way to characterize this low dimensional structure. How do we characterize the high dimensionality of these representations? The fact that we can separate these 2 points from these other 2. So we literally do exactly what I just said. So we consider all possible colorings of the points, or all possible dichotomies, all possible variables. So for instance, here we separate the cylinders from the circles, and we check whether a linear decoder can separate these 2 points from these other 2 points. The answer here is yes. So there another coloring, which is another dichotomy or another variable, and is also yes. This one I cannot. So the shattering dimensionality is going to be the average decoding performance over all possible colorings. So it's 1 when I can separate for all possible colorings, and it would be 0.5 if I cannot separate for any coloring. It would not be such an interesting. Okay, so if I do in simulations, I compute CCGP as a function of the displacement magnitude. So I start from this low dimensional disentangled representation and I distort them. You see that the CCGP goes down, but initially goes down very slowly. And the shattering dimensionality goes up. You see in these very simple simulations that, of course, are not that realistic, that there is one region here where you have a really good compromise. So this is to tell you that these 2 structures can coexist. You can still have the ability to separate all possible dichotomies, even in a representation that has a relatively high cross-condition generalization. Okay, now if I look at the paper with Silvia Bernardi in Salzman's lab, the first one that I did, and I look at the decoding accuracy for all possible dichotomies, which are all these circles here. This is in the hippocampus, the lateral prefrontal cortex, and ACC. It's always incredibly high. I can really separate every possible dichotomy. This is real data. It's cross-validated, and there's a lot of noise. And nevertheless, it's around 0.9. So this is something we see almost in every data set that we have. Maybe there is something. Okay, so now what we did recently is to analyze what is probably the only large data set that is publicly available in which the multiple labs recorded from basically the entire. Okay, so this is this huge effort of the International Brain Lab. And now, this data set is available. You can see here, every dot is a spike that is emitted during one time. Okay, so this is something we recently uploaded to the bioarchive. There are a lot of results there, but I'm going to summarize only what is relevant for the geometry. And again, Lorenzo here is the first author together with Shuzi Wong. It's a collaboration also with Liam Paninsky, who is officially in this collaboration in the IBL collection. Okay, so the task, fortunately, is a bit simple, but for good reasons. This has had to be reproduced by a lot of different labs. And essentially what they did was to put a mouse here, and the mouse controls a wheel, and then you show a stimulus that can either appear on the right or on the left. And the mouse has to turn this steering wheel to bring the stimulus to the center. If it does so, then it gets a reward. So here, this will be a wrong try. Okay, so it appears on the right and it moves it on the right, so it will actually go in the front door. Okay, the interesting thing is that at some point they change the statistics and they make the stimulus appear more likely on one side than on the other side. And then they switch which side appears. Okay, so here the task is relatively simple, but there are a lot of variables you can analyze. And also they analyze many aspects of the behavior of the animal, like whisking, for instance. Okay, so they recorded from pretty much the entire brain. We analyzed what happens in the cortex. That's the first thing that we wanted to do. So we have 43 different brain areas in the cortex. And here they're grouped, according to all these different modules. But, each of these areas has hundreds and thousands of neurons that are being recorded by multiple. Now, if you know this beautiful work by anatomist in 2019, then you realize that here you can sort all the brain areas and place them along this hierarchy. Okay, so the hierarchy is basically determined by the statistics of the incoming connections and the outgoing connections from every brain area. And you have here at the bottom of the hierarchy all the primary sensory areas, and here the more cognitive. Okay, so it's just a way to say I'm sorting the brain areas from sensory to more cognitive. And I have this order. Okay, so now I can measure this shattering dimensionality as a function of the position in the hierarchy. And here, every dot is a different brain area for which we have enough data that we can do the. Okay, again, this is all cross-validated, and it's single trial decoding for all possible different types. And you see that there is some interesting trend. So basically, some of the more sensory areas tend to have a lower dimensional representation. More cognitive areas have a higher dimensional. However, there is something you should consider. Whenever you see a low dimensional representation, this can be interpreted in multiple ways. Okay, so let me give you a couple of examples, and I'm almost done. So one possible scenario for having a low shattering dimensionality is that you have 4 different representations for these 4 different stimuli. I'm going back to this simple example. So these different points are distinct. So I can separate each pair of points without any problem. But they're all on a line, and that's why the representation is low dimension. However, there's a second scenario in which basically these 2 representations here are indistinguishable. Okay, so basically in this scenario, I'm representing only shape and not color. So one of the variables is not represented. So is this really a low dimensional representation? I mean, if you do it in the proper way, and this is an argument that was already made by Surya Ganguly and Zhao in 2015, let's say I merge these 2 conditions. These are not independent experimental conditions, so I merge them. And now I consider this new scenario in which I have condition 1 and condition 2. So it's true that in this case, the dimensionality is still 1. Okay, in both cases, dimensionality is 1. However, here, this is as high dimensional as it can be, because I have only 2 points. So this is not what I would call a low dimensional representation. Or at least, I should say very clearly that if you consider only the independent conditions, this representation is as high dimensional as it can, because there is an upper bound imposed by the number of independent conditions. Okay, so now if I redo the analysis of the shattering dimensionality by considering only the independent conditions, and we have an algorithm for merging the conditions that cannot be discriminated by linear decoder, then we get a very different picture. So this was the original one. And now this is with only independent conditions. And now you see it's completely flat, maybe with the exception of the gustatory cortex. We don't know why, but all the other brain areas are here, which means that essentially we are in the second scenario here. So if you consider only the variables that are encoded, and there are certainly more variables that are encoded in the cognitive areas than in the sensory, then the representations are all as high dimensional as they can be throughout the entire. But this is compatible with what we see in all these other studies. And this is probably the only thing I feel I can say that they have in common all the analysis that we have. Okay, so I will add this to the conclusions that I really believe that are not going to change in 10 years. Abstract disentangled representations are observed, multiple species, multiple brain areas. The geometry correlates with behavior. I showed you a few examples because we want to do more. We want to do manipulative experiments. And the shattering dimensionality is typically high, which doesn't mean that you don't have this low dimensional scaffold. You can still have that. We are analyzing that also in the IBL data, but we're still not. Okay, so now I will conclude, and this is just many of the papers that are related to this subject. We also have a lot of theory papers where we try to understand how these representations are formed if you use very simple gradient method to train a network. But I really don't have time to talk about them, but if you're interested, if you publish that you can read. Thanks a lot for your attention.
We are on the same line. But
Oh, sorry, wait, you go ahead, John, yeah.
So great talk. You talked mostly about binary variables. How do you think things change if you had, for example, 5 different kinds of shapes and 5 different colors? In particular, do you think you need as many dimensions as there are pairs of things to separate, or do you think things would be somewhat lower?
So it really depends on the kind of structure that you have. So it's not entirely true that I consider only binary cases, because the visual stimulus in the human case has 4 values, because there are 4 different stimuli. So in that case, we did the analysis by looking at 1 pair at a time, but it's clearly not the best thing. It's just that we wanted to reduce our tools. And so we can certainly do a different analysis, and we are already thinking about that. In all these cases, you can learn a lot by looking at 2 at a time. So 2 values at a time if you have a multi-valued variable. You can identify these kind of structures, but I'm sure you can do better, by developing some new. Now, if you have a continuous value variable, but you can binarize it, which is what we did. But you can also discretize it, and you can determine, what is the resolution that you can afford with that kind of data. And once you have these multiple values, you can go back to what we just discussed. One of the quantities that is very simple to generalize to multi-valued variables is the parallelism, because you can take the first 2 values, then the second 2 values, and look at the parallelism between these coded variables. So that's something we are already doing, and we basically don't need to change tools. But, this is definitely one important way.
Great. To understand the relationship between this idea of sort of a multiplex concept space and the last 100 years of development, we know that at least some representations are partitioned in this at least somewhat modular way in the brain, not just in the sense that you have neurons with more sensitive fields that are tuned to other things, but also that that regions to those parts of the brain selectively impair sensitivities. So, and it's not like these things are obviously there's not a mutually exclusive, right? But I'm just wondering, what is the kind of the theoretical perspective on this? Like, why do we have both of these?
Okay, so I don't know if I have an explanation for that, but in this study of the IBL dataset, what we see is that it really depends on the scale that you're considering. So if you're considering the entire brain, it's certainly not true that everything is everywhere and you see a modular structure. To the point that you give me the response profile of 1 neuron, I can tell you with a decoder to which brain area it belongs. because they're different. The statistics is different. I mean, there's a lot of diversity within each brain area, but they're different across different brain areas. However, when I look at one particular brain, so now I zoom in into a particular brain area, I don't see this pattern. And I don't know whether it's due to very strong recurrent connectivity. I'm only talking about the cortex now, or for some other reasons, but we just don't see it at that scale. So I don't have a solid explanation for this. It's something we see very often if we simulate a recurrent neural network with very high recurrent connectivity. Typically, I mean, we simulate fully connected recurrent network. The structure that you have in the input is lost very easily. So you might have these very nice disentangled representations in which you actually have highly specialized neurons in the input, but you don't see it in the recurrent. And we definitely need a theory to better understand.
Thank you. So I'm curious about the temporal order by which these features separated over the, I mean, for example, over the task. So for example, I can imagine the context first, like bias neural activity to go in two different like subspace and then the other feature separated. Do we know anything?
It's a much more difficult analysis because then you need to consider a much shorter time interval. Because if I understood correctly, you want to know what happens during one trial. So you look at the time course of many of these quantities of all trials. So in all these studies, we did a little bit of that, but we could only look at the time interval preceding the stimulus and the time interval during the stimulus. We can look at how some of the quantities are encoded during the trial. And I can tell you, at least in the monkey case, we could do this analysis. And you can see a couple of things. So first of all, there's one very interesting observation, which is that you can decode the stimulus identity for many different brain areas. And typically you do it at different times. Okay, so the amygdala is the first one where you can identify the visions. When you try to decode the decision of the monkey, and here we can disentangle the two because we have this complex, that decoding accuracy goes up almost exactly the same time in all the brain areas that we reported from, with only one exception, which is the hippocampus. The hippocampus is significantly late. So it looks like there is some kind of global decision that is broadcasted to the hippocampus. So the decision is not made in the hippocampus, although the hippocampus might encode information that is relevant for making the decision. But at least this is what we see in the monkey.
Yeah, thank you for that careful talk. Today, I guess like most of the study was in most of the studies, animals and humans focus on just one thing at a time. So in other words, like they care about the one problem. But in the reality, there are multiple objects in the visual field in the world. So like in the earlier slide, like if it's multiple objects present in the slide, and then for example in the visual search task, they have to construct this geometry in each part of the visual field to parse out like what's where. And how do you think the brain's going to solve this?
So I don't know, and we never analyzed these kind of experiments where you would have multiple objects on the screen. I can tell you a couple of things. So the first one is that if you use perfectly disentangled representations, you're going to have the binding problem. Okay, so disentangled representations are bad for memory capacity, binding problem, and for separating, for flexibility, for separating the points in any way you like. They're good for generalization, but they're bad for all these aspects. So you must have somewhere some nonlinear distortion that are really significant. And I would love to see more, in this respect. One of my postdocs, Jeff Johnston, did this study with Ben Hayden, and they actually see these kind of structures. The second thing is that there are probably attentional mechanisms. And so it's very possible that, at least in some brain areas, you really see one at the time. even if you don't move your eye. You have some covert effect. But we've never analyzed this data, so I don't know what happens.
There's increasing numbers of papers showing that concepts are encoded on grid maps, not just space. So I'm wondering, how do you think of your geometry framework in relationship to the grid maps?
The concepts are encoded.
Encoded on like the hexagonal grid structure, not just space in grid cells.
Oh, grid cells. Yeah. So, I mean, grid cells, is an interesting, relatively low dimensional structure. And I'm sure that it plays a very important role for memory, for organizing the memories. And Lafitte has this paper that just came out in Nature that shows how to take advantage of this structure to store efficiently an exponential number of memories. But we never thought specifically about grid cells and how they could be incorporated into this theoretical framework. And it's certainly something interesting. It's a relatively low dimensional structure, so it's a non-obvious structure. It's not something you would get by chance. And so that's why it's interesting to look at it. One of the problems is that we have some data, and typically the grid cells are a minority of the cells, so it's very difficult to do the analysis. And there are less than 10% of the cells in the parameter.
When looking at analogous regions across species, do you see that like the same ones in analogous regions share that like characteristic of transforming across the transforming representations, parallel action? And also like, do you have hypotheses as to why like ACC wasn't able to, or not wasn't able to, but it didn't have that parallel action?
So we don't know. So the only case in which we could monitor the formation of these representations is the one that I showed, because humans can learn this in 1 second. And so that's the only data set that we could analyze and do this. There are clearly differences when you consider different experiments or different species. So the same experiment in humans and monkeys, we have that in monkeys, we see that probably all the important things happen in prefrontal cortex, particularly dorsolateral prefrontal cortex. In humans, we don't have dorsolateral prefrontal cortex, but we see that the only interesting place where we can observe this phenomena is the hippocampus. In all the other brain areas that are listed at the beginning, we don't see the formation of this. Now, one possibility is that humans, of course, are different from monkeys, but the most likely explanation is that monkeys are overtrained, whereas humans learn only in 1 session. So it's possible that in monkeys, we are already in a regime where the hippocampus doesn't play an important role any longer, and everything has been transferred to the yeah.
It's a really nice visual experiment. What's the new amount of neurons that you need? So if you reduce the number of neurons, can you go down for the same results with the 1/3 of neon and under the.
So in the case of the Bernardi dataset from Salzman's lab, somebody in Salzman's lab did this analysis, and it turned out that in that case, 40 neurons was the mean to do this analysis. Now, it also depends on the brain area you're considering and the species and the way you're recording the activity. So if you have electrophysiology is one story. If you have calcium imaging, then you need many more neurons because the number of events is much smaller. Also, in human brains, the firing rates are typically lower than in monkeys. So you probably need more than 14. And so, yeah, it depends on a lot of factors, but you don't need an enormous number of neurons to do it. The main limitation is that we want to use only cross-validated quantities so that are computed on the performance of decoders in single trials. And so you need a minimal number of neurons, but it's not an unreasonable number of
There are no more questions, which thanks Stefano.